Life in 4 letters, structure and function of DNA. Table of contents. 1. Introduction to DNA. 2. Replication. 3. Genes and chromosomes. 4. Genetic inheritance. DNA, or deoxyribonucleic acid, is an essential molecule found in the nucleus of living organisms' cells. It can be compared to a book of genetic instructions that determines how an organism develops and functions. Early mentions. During the mid-1980s and early 1990s, scientists explored trait inheritance in organisms through experiments with peas and flies, laying the groundwork for understanding control mechanisms. Key discovery. In 1953, scientists discovered the double helix structure of DNA, using data from other scientists. This revealed how DNA stores genetic information. Significance. The discovery of DNA's structure revolutionized biology and genetics, allowing for a better understanding of inheritance and evolution. From a chemical perspective, DNA is a polymer of nucleotides, in other words, a polynucleotide. A polymer is a compound formed by many simple units connected to each other. Double helix structure. DNA resembles a twisted ladder with 2 strands containing groups of 4 nitrogenous bases, adenine, A, cytosine, C, guanine, G, and thymine, T. Base pairing. The bases are paired in the helix, specifically A pairs with T, and C pairs with G. Nucleotides are the basic units of DNA, composed of a phosphate group, P, a sugar, deoxyribose, and a nitrogenous base, A, C, G, or T, that encodes genetic information. Information storage. DNA stores the genetic information that determines the characteristics and functions of an organism. Replication. DNA can make exact copies of itself, essential for cell division and growth. Transcription and translation. DNA is transcribed into RNA and then translated into proteins, controlling cellular activities. Inheritance. DNA is inherited from generation to generation, passing traits from parents to offspring. Replication. DNA replication is the process by which the DNA molecule is duplicated before a cell divides. It ensures accurate inheritance and is essential for cell reproduction. 1. Strand separation. The 2 DNA strands unwind and separate, breaking the hydrogen bonds between complementary bases AT, CG. 2. Complementation. Free nucleotides pair with the exposed bases on each strand, following the rules of complementarity. 3. Formation of new strands. 2 new DNA strands are formed, one for each original strand, using the paired nucleotides as building blocks. 4. 2 DNA molecules. The result is the formation of 2 DNA molecules, identical to the original, ready for cell division. Accurate DNA replication during cell division ensures that each daughter cell receives an exact copy of the genetic information, preventing genetic errors and cellular dysfunctions. DNA forms genes, and multiple genes are grouped into chromosomes, present in the nucleus of every cell, containing all the genetic information of an organism. Genes and chromosomes. Chromosome, structures formed by DNA and proteins housing multiple genes in the cell nucleus. Genes, segments of DNA containing instructions for protein synthesis and determining hereditary traits. Genetic inheritance. 19th century, experiments with peas revealed patterns of inheritance, though the connection to DNA was not known. 20th century, DNA's role as the carrier of genetic information was discovered, revolutionizing genetics and biology. Present, DNA sequencing and genetic engineering enable deep study of genes and their influence on inheritance. The transmission of genetic information, including DNA or RNA, determines the continuity and diversity of species on Earth, contributing to the inheritance of physical traits and behavior patterns. Inheritance, transmission of genetic information from parents to offspring, determining characteristics in the progeny. Diversity of traits, inheritance perpetuates and diversifies biological traits in all species. Continuity of species, essential for species continuity and adaptation over generations. Do you have any questions?